So DNA replication, basically we need to understand how DNA replicates. So DNA has to replicate, it has to produce more DNA to replace basically dead or, I don't know, messed up DNA that needs to be replaced. It needs to make its own DNA. So DNA needs to replicate. Every cell in our body has this DNA in it, and this DNA, we're making new cells in our body every day, so we've got to create DNA to be able to, be in place inside of the cells. So before an organism can grow, the DNA must be copied. And it says, remember what happened in mitosis. So remember that in mitosis, you start with one cell and you end with two. It sort of like creates a clone. That's where DNA replication is going to happen. So in mitosis is when DNA is going to replicate to make that second new cell. All right. So here are the steps, basically, of what happens when DNA replicates. And I'm just going to go through this slowly. I just wanted to make sure that this wasn't cut off. Do we have... Well, we'll figure it out when we get there. So the very first thing that happens when we're replicating DNA is that it untwists. And so when we look at our DNA, it's like this. It's really twisted up. So it needs to untwist so that it is sort of like a ladder. Two pieces stuck together, okay? So once it untwists, the next step is it's going to unzip. So if it was to look like this, well, what it's going to do is it's going to sort of look like this. So some of these pieces are going to be connected, and then some of these are going to be sort of looking like you're opening a zipper, if that makes sense. And why it needs to do that is it needs to open it up so we can create pieces that will go here. We need to create pieces that will connect to those nucleotides, those open nitrogen bases, put their complementary there so that we can create a new set of DNA. All right, so that's what happens in that step. So untwists, then unzips. Then the third step is that the free-floating nucleotides will attach to the exposed bases. So what that means, well, so if we have our open zipper of our DNA, and it's unzipped, then we are going to, there's going to be free-floating nucleotides. So nucleotides, remember, are a phosphate, a sugar, and a nitrogen base. So those pieces are going to be floating around in the nucleus where this DNA replication is happening, and they are going to find and bond to here. So they're going to actually bond. So let's say this is A, T, G. So these free floating, those are the originals. And then these are the originals. Oops. So they're unzipped, And then these free-floating nucleotides, like, I'll make them in red. They're just floating around out here. And they could be all different types. They could be G's, they could be A's, they could be T's, they could be C's. They could be all the different types. So basically, they're going to bond to this open one. So these ones were once bonded, but now we've unzipped them and we have bonded them to, we're bonding them to these new ones. So now this C will move in and bond there. This T will move in and bond there. This A will bond there, and this G. That looks pretty confusing. But that is how it works. So they will all match up. So this guy, I'm just going to draw one of them, will go there. So we end up with the G bonded to a C. And it's a new C. All right. And then as this is a very simplified version, but once all of this has been unwound, unzipped, then there's two pieces or molecules of DNA, and then they can coil back up. So let's say we started with ATGC, TACG. Then we would end up with that, basically another piece of it after it replicated. All right, so we start out with one piece of DNA, which is this, and we end up with a second piece of DNA. Okay, and it's exact copy of the original one that we started with. We call this very first one that we started with the template. So this is the template strand. And then the piece that we create is known as the complementary. So I'm going to write this one's the template. And then that's the complementary. Okay. So that's how, a very simplified version of how we might look at DNA. Oops. So here's a picture of it, better than my drawings of it. It says DNA and twists and bands between the nitrogen bases are broken. Then free-floating nucleotides will attach to the exposed halves of the DNA and produce new DNA strands. So we started out with one twist, and now we've got two. We've got this one and this one, okay? So that's how we replicate DNA. There's a video there we can't watch. I will put it on the Google Classroom. We can't do this activity that they showed, but I will put up a video of how this works, but we're doing it pretty in a simple way, I guess you could say. And that's really all that you need to know about DNA replication, I guess, at this time. And then we'll stop there right now, and then the final lesson we'll learn about protein synthesis.